Hello, everybody, and welcome to today's live broadcast. We're talking on the topic of rapid PGS workflow using NGS sequencing, and the two speakers today are Dr. Fabien Murisier and Dr. Luis Alcaraz. I'm Colin Davidson with Thermo Fisher Scientific, and I'll be your moderator for today's event. Before we begin, I'd like to remind everybody this is an interactive event, and we encourage you to participate by submitting as many questions as you would like at the time you want during the presentation. All you have to do is click the green question and answer button located in the lower left part of the presentation window and type the question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of today's presentations. Also, please note that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have any trouble seeing or hearing this presentation, please click the support button on the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. I'd like to introduce our two speakers. Fabien will be our first speaker. He graduated as a biologist at the University of Lausanne in 2002. He then obtained his PhD in 2006, after which he joined the Human Genetics Unit at the MRC in Edinburgh, where his research focus was on the genetics of embryonic development. In 2007, he joined the Laboratory of Reproductive Biology for the Center of Medically Assisted Procreation, at which he acquired his ESHRE certification as a clinical embryologist in 2010. Since 2013, he is responsible for in vitro fertilization lab of Fribourg, Switzerland, and Dr. Murisier is also the scientific director of the Fertas laboratory since 2011. Our second presenter is Luis. He's a technical manager and co-founder of ESHRE. He developed his scientific career in many different fields related to molecular biology, from gene sequencing to drug discovery, but always with a strong interest in bioinformatics. He obtained his PhD degree in proteomics. During his postdoc, he changed the course of his research to develop biosensors, working on a project to detect biomarkers in culture media of human embryos related to aneuploidy. At this time, he funded ESHRE with Andreas Anton, and from this moment, Luis has been working passionately towards applying microarrays and next-generation sequencing to genetic diagnosis. And with that, I'll turn over the audio control to our first speaker, Fabien.
So good morning, everyone. So we'll switch to my presentation. So as Colin properly introduced, I'm an embryologist, not a geneticist, and I will present you some of our recent work on pre-implantation analysis. I have to say that in Switzerland, the law has changed very recently. Last year, PGD and PGS were forbidden. And since a few months, we are allowed to propose PGD and PGS to our patients. And linked to this big change in our country, we have developed some program of analysis in order to implement it. So if we go back to the history of pre-implantation genetic screening, in the early days, it was done by FISH, so fluorescent in situ hybridization, on cleavage-stage embryo. And at that time, the number of chromosomes that could be analyzed was limited due to the techniques. There have been other technical issues such as cell fixation problems, and the specificity of the FISH probes was not that high, at around 80–95%. And they have discovered at that time that the embryo at 8-cell stage is highly mosaic, which means not all cells have the same chromosomal contents. Another discovery that was made at that time is that embryo biopsy on the 3 embryo or 8 cells embryo impacts the embryo survival. And the outcome, which means if we do genetic analysis of embryo, does it improve the chance of pregnancy during an IVF cycles? And in this Cochrane review, they have shown that it was better not to do PGS on cleavage stage embryo, because the damage you do to the embryo was worse than the benefit of the genetic analysis. So it was somehow disappointing results for the early stage of PGS. And then came out more advanced techniques, which allows to analyze not only a few chromosomes, but the whole chromosomal contents of a few cells, all 24 chromosomes. And there have been several publications showing that it was feasible to do a comprehensive analysis on early embryo. And it was also shown that previous fish analyses were not really reliable. Then another revolution came out in our field, which was doing the biopsy not anymore at day 3, but at day 5, so at the blastocyst stage. And the reason why it's somehow a revolution first, it's because it's safer for the embryo. As you can see on the right side, on that graph, they have compared here, it's a cleavage stage embryo biopsy, and here you see the blastocyst biopsy. And as you can see, when doing a biopsy at day 3 embryo, the implantation is decreased, which means the biopsy impairs embryo development. Whereas when the biopsy is done at the blastocyst stage, it's very similar, which means it's not harmful for the embryo or rarely harmful. The other benefit of doing blastocyst biopsy is that you get more cells for genetic analysis, not only 1 or 2, but 5 to 10 cells, which mean much more accurate results. It's also a cost-efficient strategy because you need to analyze less embryo. Because it's well known that more or less half of the embryo will not develop to the blastocyst stage, which means some of them will naturally arrest in the development, which means the one you need to analyze is much lower. And as you can see on this graph, you have twice more day 3 embryo compared to blastocysts. And this is even at lower number when the maternal age increase. And the target patients are around that area, which means you have like 4 or 5 blastocysts to analyze instead of 8, 10 day 3 embryo. So it's twice less genetic analysis, we could say, on average. Another revolution was the vitrification of blastocysts, which leaves enough time to do the genetic analysis. So in this publication, this revolution, which is a combination of comprehensive whole chromosome analysis and blastocyst biopsy, was named PGS 2.0. And at that time, 1 year ago, it was seen as the future of PGS, and it has been said that all the previous results coming from fish analysis should be forgotten, and we are entering into a new area that is entirely different from the previous one. And these are the outcomes. I've taken 2 prospective randomized trials here, and as you can see on the left side, in this study they have compared untested 2 blastocyst transfer, which is in yellow here and here. They have compared it to a single euploid blastocyst transfer, the blue blocks. And as you can see, when applying PGS to in that situation, you have at least the same pregnancy rates as transferring 2 blastocysts without test. So this means without any reduction in efficiency in terms of clinical pregnancy, you were able to avoid having twins. In the other study by Scott et al. On the right side, they have shown that the implantation rate was improved by PGS 2.0. But then come out another issue which was not visible or not that well known before, is that embryo can be mosaic, which means not all cells of the embryo have the same chromosomal contents. Some cells, which are on the top represented in yellow, such as here, have abnormal chromosomal contents, either chromosome in excess or chromosome missing, whereas other parts of the embryo, like here, have a normal chromosomal contents. So depending on where you do the biopsy, you might end up, if you do the biopsy here, you might end up with a fully normal embryo. If you do the biopsy here, you might end up with a mixture of cells, normal and abnormal cells. And this is well seen on the graph here during the analysis, where you see here that there is a mixture of normal and abnormal cells. This would be fully abnormal, so one chromosome missing. This would be 2 copies of the chromosomes, and this would be 3 copies of the chromosomes. And in that case, it's in between, which means there are some cells that have 1 copy and some cells that have 2 copies. This is easy visible. This means that it's around 50% of cells that are abnormal. Whereas on the bottom here, you see it's not that clearly visible. It's most likely that only 20% of the cells are abnormal. So this is somehow an issue depending on where you do the biopsy. The advantage of doing a blastocyst biopsy, as shown here, is that as you have more cells for the analysis, you have also more accurate results, and you are most likely to detect mosaicism. So a big question when scientists have discovered that mosaicism in embryo is quite common is it how frequent it is? And if you read the literature, there are many differences and many discrepancies. They've seen from 10 to 90% mosaicism in day 3 cleavage stage human embryo, and the mosaicism in blastocyst has been shown to be present in 5 to 50% in blastocyst. So as you can see, the variability is quite high, and some explanation of why those differences between the different publications could be linked to technical aspect, number of chromosomes analyzed, technique used. It could be also linked to classification criteria, what is defined as mosaic or not. It could be also linked to the patient group analyzed in the study, for depending on the stimulation protocol. It has been shown that some stimulation protocol might, or some culture condition might affect the percentage of mosaicism, and also the type of samples that have been used for the research project. Is it arrested embryo? Is it donated embryo from egg donors? Or it depends from publication to publication. And then it also depends on the analysis method. Some of them are more accurate to detect mosaicism. So then come out a new revelation for PGS, which is next generation sequencing, which has been shown to be at least as efficient as array CGH to detect and to quantify the number of chromosomes in a pre-implantation samples. And in that study, they have shown that the implantation rate is almost doubled when comparing to the control group, which means it's physical and efficient to screen for aneuploidy in embryo using NGS. And one big advantage of NGS compared to the more conventional array CGH method is that the dynamic range of NGS is much higher compared to array CGH. As you can see here, this is array CGH results. You see that there is an extra chromosomes, but the dynamic range is much higher in using next generation sequencing. And in that case, it's a trisomy of the chromosome 7, which is clearly visible here. Which is also visible here on the array CGH, but to a lower extent because the noise is taking is getting quite big. And then if you look at mosaic samples, so in that case there is also a mosaicist for chromosome 12, as shown here by next generation sequencing analysis, where there are around 50% of the cells that have an extra chromosome 12. If you look at array CGH data, it's much less visible. It's very close to the noise, so it's difficult to say whether it's noisy or whether it's mosaicism. So this is one big advantage of NGS analysis. It allows a broader range of analysis, and it allows to see mosaicism much better. So I think we are entering into a new area of PGS, which we could term PGS 3.0. And this has a couple of premises. First, it allows an accurate detection and quantification of mosaicism, even at low amount of mosaicism. It also allows to detect not only full chromosome aneuploidy, but also if some part of the chromosome is missing or part of the chromosome is duplicated, what we call segmental aneuploidy. And even it allows to identify the combination of both, which is mosaic segmental aneuploidy, and it has a promise of lower cost. There are still some open technical questions. What is the limit on the accuracy of detection? How down to the percentage of mosaicism can we go? Are we able to detect small deletion or small duplication? And what about the multiplexing capacity? Are we able to analyze simultaneously many samples? And based on all that question, when we decided to start with our PGS program, we have done some preliminary studies, which I will show you right now. So basically, the technical procedure is this one. So you start with the embryo biopsy of the trophectoderm at day 5, and you get around 5 to 10 cells. Then you do whole genome amplification to massively amplify the amount of DNA you have. And then you submit this amplified DNA to next generation sequencing. This allows you to generate or to sequence many different very small sequences, thousands of small sequences from your pre-implantation samples. And then with the miracle of bioinformatics, the system is able to align all these small sequences to the reference genome. And by counting the number of each small sequence, you are able to quantify the number of chromosomes you have in your sample. And as you can see here, for these chromosomes, there are not enough sequence compared to what is expected, which means it's a monosomy for these chromosomes. So we have compared different methods and then decided to go for the ReproSec PGS kits run on the S5 systems. And as you can see here, some of the big advantage of the technique is that it's a fully standardized, ready-to-use kits, with a huge option of automation of the procedures, together with option to have different size of sequencing chip, 16 samples, 24 samples, and up to 96 samples. So it adds flexibility and automation, which we have seen as a big advantage. And then on the top of that, there is a strong bioinformatic program which allows to detect mosaicism and to add some options such as gender masking, because it's, at least in Switzerland, it's banned to discard the embryo gender. And it's quite quick. Everything in our end could be run in less than 12 hours. even if we had some optional controls, quality controls. So this is our study design. So we had 3 types of samples for our test. We have been using cell lines, which are chromosomically well characterized with deletion and duplication. We also have used our own prenatal samples, so from patients doing prenatal analysis in our genetic labs. These are those 4. And with the cell line, we have also created artificial mosaic samples by mixing those samples or those samples to generate artificial mosaic, and we have access to a few but some developmentally arrested embryo from patients. And these are some of the results we get. So maybe let's start first by explaining you what you have to look at and what those graphics are. So on the bottom you have, starting here, you have all the chromosomes in a row. So this is chromosome 1 down to chromosome Y here. So you have all chromosomes that are lining on the bottom, and here on the right you have the copy numbers. So in the middle here it's 2 copies of the chromosomes. Here it's 1 copy, and here it's 3 copies. So as you can see in this sample, for all chromosomes starting from 1, you have 2 copies, with only one exception, which is here, where you have 1 copy because it's a male embryo or male cell lines with 1 copy of the X and 1 copy of the Y. So in this first study, what we have done is we have run 96 samples together, and we would like to check whether it was feasible to see aneuploidy of a small chromosomes. So it was chromosome 21, trisomy 21. And as you can see here, we are able to see trisomy from the DNA equivalent of 1 cell. And this is also, it works very well for 5 cells, 10 cells, and 20 cells. So we were really reassured by doing that, which means even when you analyzing 96 samples in parallel, it's feasible to see a trisomy 21 at a concentration corresponding to only 1 cell. Then the next test we have done is to see whether we are able to see subchromosomal deletion or duplication. So in that case, we analyzed 24 samples at the same time on the smaller chip, and we were happy to be able to see a 10 megabase deletion chromosome 2, which is shown here, and we were also happy to be able to see a 21 megabase chromosomal duplication. And we are at the equivalent of 2 to 3 cells DNA amount for both, which means this techniques allows to see subchromosomal deletion and duplication very accurately, and as you can see, there is almost no noise. Then the question was, is it feasible to see deletion or duplication when analyzing more samples together? In that case, it's 96 samples analyzed together, and as you can see, even at the equivalent of 1 cell, the deletion is well visible there, which works as well when you have more amount of DNA. So even when analyzing 96 samples together, the detection efficiency remains excellent. Then we try to analyze whether with this technology it's feasible to analyze mosaic samples. So in that case, we have done a mixture of cell lines which carries 1 X copy and two copies of the X for the other part. So this is what you see here. So in that case, it's 0% mosaicism, and then we go up 20, 40, and up to 80. And as you can see here, you can see the mosaicism increasing with a very quantitative manner. So you see that the shift between the 2 copies line and the analysis is pretty linear compared to the percentage of mosaicism. And in that case, it is still 26 samples analyzed together with a DNA amount of 5 cells, which means for 20% it means only one cell is different from the others. So it means that you are able to, if you have 5 cells, you are able to see a discrepancy within only 1 cell. Then the last challenge was to put everything together, whether it's feasible to analyze mosaicism, which is segmental. So mosaicism when only part of a chromosome is missing. And we are somehow pushing the techniques at its limits. So we keep analyzing 96 samples. And as you can see here, it's feasible to see segmental mosaic aneuploidy as shown here and here. So this was our last experiment, and we decided to push the system to its limit, and it's still quite good in terms of results. Then we have run a couple of real samples, which are arrested embryos that have been donated by the patient, which means that these are embryos that did not go to the blastocyst stage, which so we will not implant, but as they were planned to do the analysis with the agreement of the patient, we have done some analysis. It also helps the patient to understand why the embryo arrested. So in that case, in this example, it's a monosomy of the chromosome 2, as shown here. And in that case, it was an embryo that didn't do any compaction, so it even do not go to the morula stage. So we split it in 2 parts and analyze the 2 parts separately from the same embryo. And as you can see, it's a monosomy of the chromosome 22, as shown here. And we get the same results for the second half of the embryo, which was reassuring. So then, regarding to the power of such a method, it comes some biological question. We are able to see many things in those embryos. We are able to see mosaicism. We are able to see subchromosomal defects. How should we deal with that, and what shall we do with that? It has been shown in some publications that mosaic embryo can implant, even if the pregnancy rates are much lower, but it's not 0. And some mosaic embryo are able to give rise to healthy baby. This could be due to discrepancy between the placenta and the fetus itself. As we are analyzing trophectoderm cells, so not the cells that will form the future fetus, but we are only analyzing the cells that will form the placenta, there could be a discrepancy where an embryo is diagnosed abnormal where it's normal. And it also been shown that, even it was more in day 3 embryo, that there are some rescue possible, which means some of the abnormal cells can be removed by the embryo. I have to say that all those things are quite rare events. It's not that often that an embryo is rescued or that there are fetal-placental discrepancy, but it's good to know that it could happen. So for my last slide of conclusion, I think we are entering into a new area of PGS. We are now able to detect segmental aneuploidy, and we are now able to detect low level of mosaicism. The question remains, shall we go to higher resolution in order to see more, or shall we reduce the resolution in order to do more at the same time and thus reduce cost for the patient. So it's also always a discussion between cost and resolution. And maybe the solution is somehow more customized. For some patients, the resolution is important. Sometimes there might be carrier of a deletion or duplication that is quite small, or you need to look for other things. And in other cases, it's also important to look at the cost. So altogether, it means that we get more information from our samples, but it means also very high complexity for the interpretation. And we are lucky to be helped by some excellent guidelines, one from the COGENE and one from the PGDIF, where you can find many recommendations, in particular on which mosaic embryo are suitable for transfer and which one are dangerous to transfer. And they are quite often revised. But the main conclusion would be that I think all those information should be well discussed together with the patient, first in preparation, so before we do the analysis, but also after, as soon as we have the results, because the final decision has to be taken by the patient, and we are here to help them, but we are not here to decide for them. And for my last slide, I would like to thank people involved in the work. So there were people from our Center for Reproductive Medicine in Lausanne, in Switzerland. It's CPMA, and then also Fertas, which is our sequencing laboratory, together with our partner MCL, our genetic laboratory. Thanks as well to Thermo Fisher for the collaboration, and I leave it to Colin for the next speaker.
Yeah, thanks so much, Fabien. So we can now transition to Luis and his presentation on PGS and PGD combined.
Okay, so thank you, Colin, and thank you to all of you for being here. My name is Luis Alcaraz. I'm technical manager at ESHRE, which is a genetic lab located in the southeast of Spain. In my company, we do almost everything in genetic analysis through leading technologies such as microarrays and next-generation sequencing, applied to prenatal, postnatal, and preimplantational genetic diagnosis. Everything using a variety of platforms. So probably you all know that in genetics we are living our golden age. Technologies are evolving incredibly fast, especially in the field of next-generation sequencing. We are living a situation similar to the computer revolution in the 80s, to such an extent that if you want to have in your lab the latest NGS technology, you must change your sequencer more often than your mobile phone. But fortunately, this is not always necessary. As you will see in the following slides, you can do very nice things with simpler sequences. Anyway, now it is possible to adapt almost any genetic test to next-generation sequencing, as we did for PGD and PGS a few years ago. We started this evolution more than 3 years ago, first for PGS with a protocol developed by ourselves, and now using ion ReproSeq. And at the same time, we made this evolution also for PGD, for monogenic disorders. And more important, we were able to join both protocols and to do both things at the same time. Moreover, I think we are probably the first lab in the world doing everything in reproductive genetics with next-generation sequencing. It doesn't matter if we are talking about PGD, PGS, translocation, monogenic diseases, or the biopsy in day 3, day 5 with fresh or frozen cycles. We always run next-generation sequencing. So, why we decided to start this movement despite being difficult? I used to say that a PGD lab is very complex. If you want to do PGS, you need to set up a lab equipped and designed to do microarrays. including a microarray scanner and a hybridization oven. Then if you want to do monogenic disorders, you need first Sanger sequencing, which includes a capillary sequencer that is most expensive than some NGS machines, and also some equipment to do fragment analysis, because you need to run the informativity test and the indirect testing, like for example an electrophoretic system. And finally, if you want to use carrier mapping, then you need other microarray scanner, but bad luck, because it's different from the scanner used for array CGS. So at the end, you need a lab that is similar to this, with a lot of equipment and a lot of people working at the same time with the same samples, all of them well coordinated by a lab manager, because we need to fine-tune all the experiments, especially when working at day 3 with fresh transfer. And this is, as you can see, this is very nice. It's wonderful, but the problem is that it's very expensive. However, next-generation sequencing has been a game-changing technique. Next-generation sequencing is like a one-man band. NGS means cost reduction, the possibility of analyzing very challenging samples, to combine different analyses, everything faster, and with an equipment that can be managed by one person without too much stress. And for this reason, in the recent years, we are trying to do everything with NGS. As you will see, with NGS we are able to do aneuploidy testing for PGS, as Fabien has shown, the analysis of point mutation for PGD, and the analysis of polymorphisms for the indirect test. Well, for PGS, for aneuploidy screening, we rely in ion reprosec solution. I'm not going to talk about it because you have seen a very nice presentation from Fabien. We use this system because it's fast, robust, and cost-effective. But what about monogenic disorders? In monogenic disorders, this is the most common situation. Usually, we have a couple. Usually, we have a couple in which both are healthy carriers of a recessive disorder. This couple is at risk of having a child affected by this disorder, and very often the couple already have a child with the disease, and they want to avoid the possibility of having another one. For this reason, they go to an IVF center to perform a PGD cycle to select the embryos without at least one of the mutations. As you know, performing PGD in a classical way requires to combine several techniques like mini sequencing and fragment analysis. We use the first one to direct detect the mutation of interest, but we need also to use an indirect test to confirm the results and exclude something that is called allele dropout by analyzing several polymorphisms around the mutation of interest. So the direct test is easy. We just sequence the mutation of interest. So imagine that these are the two alleles of a gene that you want to sequence, and in red we have the mutation we are interested in. Usually after PCR we have several copies of both alleles, and when we sequence we can see the mutation by this way, because we can see a mix of two peaks. So according to this result, we can classify this embryo as a carrier of the mutation. So that's easy. But very often, around in 5% of the cases, we have a phenomenon called allele dropout. This is caused by a technical artifact in which during the PCR only one allele is amplified and the other one is missed. So when we sequence, we get the results from just one allele. In that case, for example, the healthy allele. The problem here is that if we look at this result, we may think that this is a healthy embryo and suitable for transfer. which is obviously a misdiagnosis. So how can we be confident in our results? So to be confident, we do the indirect test. For the indirect test, what we do is to look around the mutation we are interested in to find polymorphisms that can help us to confirm the results. So here, for example, we have chromosomes, and here the mutation we are analyzing. What we do is to look around the mutation for a special type of polymorphisms that are called short tandem repeats. In STRs, we have a short sequence that can be repeated several times, and the key point here is that any of us have a different number of repetitions. So by analyzing the couple and other affected family members, we are able to know which polymorphisms are shared between all the affected family members. So if we don't see the mutation of interest, neither any polymorphism shared with the affected family members, we can be confident in our results. So for this reason, this classical way of doing PGD is expensive and annoying for the couple, because usually we need samples from other family members in order to complete the informativity test, and this requires time and money. Moreover, usually couples don't combine PGS and PGD because this increases the price quite a lot, since we need to combine different techniques. So for this reason, after working for a while with PGS, we decided to try to do also the study of monogenic disorders. So when we decided to make this movement to NGS, in the market there were a couple of platforms offering benchtop sequencer. At this point, we decided to go for the ion Torrent machine for this reason. The first one is that it's fast, since it is possible to complete more than a run in a day. The second one is that it's flexible, because you can use 3 different types of chips with different throughputs. So we can process few or many samples without affecting so much the price. And finally, this technique is very cost effective. So with NGS, the direct test is so with NGS the direct test is easy. We just need to sequence the region we want to analyze. Here, for example, you have an example. This is a couple in which he is carrier of a point mutation in the NF2 gene, which is responsible of neurofibromatosis type 2. You can see here the mutation, and as you can see, there is no mutation in the sample of her couple. So, but the indirect test is a little bit more difficult. It's not possible to use STR for the analysis, because STR are quite difficult to analyze by NGS for technical reasons. But we have other possibilities. So we decided to analyze several polymorphisms, 1 megabase close to the mutation, but we use SNPs instead of STR. The problem with SNPs is that even in 1 megabase, it is possible to find thousands of SNPs, but most of them won't be informative, because both parents will be homozygous for this polymorphism. So the first challenge is to select the SNPs more likely to be informative, which is not easy. But we developed an algorithm to do this. This is quite complex and takes into consideration different things, as for example the frequency in the population. Here, for example, you have a graphic in which you can see the different distribution of different SNPs depending on the population of interest. Other thing to consider is the distribution in haploblocks. An haploblocks is a set of SNPs that are inherited together. The problem here is that if you select several SNPs and all of them belongs to the same haploblock, this is a waste of time, because all of them will work in the same way. So if one is non-informative, none will be. So at the end, what we do is to select more than 100 SNPs around the mutation that are more likely to be informative. To amplify all these SNPs, we use the AmpliSeq technology. AmpliSeq is a great tool for this because it's a highly multiplexed PCR. With AmpliSeq, we are able to amplify thousands of PCR products in one single reaction, and this simplifies the protocol. So, if we want to combine PGS and PGD, we do low coverage sequencing, as you have seen in the Fabien presentation for PGS, and for PGD we do an enrichment of the region of interest with AmpliSeq technology, which allow us to have high coverage there. The protocol is also very simple. First we do the amplification with the ion reprosec kit. Then we take an aliquot of the amplified material and we perform the amplification of the mutation and the selected SNPs with AmpliSeq. We put together both libraries and perform the templating with isothermal method. And finally, we do the sequencing with 100 VSPR and the bioinformatic analysis. This is the manual protocol with PGM, but it is also validated with IonChef and S5. For the bioinformatic analysis, we follow two ways. First, for PGS, we use the ion reported as recommended, but for PGD, we follow a custom protocol, which consists first in to perform the alignment of the reads and the variant calling with the software provided in the ion server, and finally we analyze the sequences to find the mutation of interest and also the SNP for phasing. And this is an example. Here you have the same example as before, in which we have a couple with the male affected by a deleterious mutation in the NF2 gene. You can see the mutation here, and if we analyze the embryos, we found that embryo number 2 and number 3 are carriers of the mutation, and number 1 and number 4 are healthy. However, if we analyze one of the informative SNPs close to the mutation, we can see that this SNP, which co-segregates with the healthy allele, is present only in the embryo number 1 and not in the embryo number 4. So that means that embryo number 1 is really healthy, and in embryo number 4 we had an allele dropout, and therefore it's not really healthy. But how we know that this SNP co-segregates with the healthy allele? We know this thanks to the SNP phasing, and I'm going to show you how it works. Imagine that we have a situation in which we have a couple carrier of a recessive disease. For SNP phasing, we always need another sample from another family member, or as in this case, a sample from a previous abortion. Imagine that these bars represent the alleles from the male patient, and this bar from the female, and obviously in the fetus we will find also two alleles, one from the father and another one from the mother. These alleles are also ligated to the pathogenic alleles in both parents because the fetus has inherited both mutations. And what we want to know is the status of each embryo, if they are healthy or not. Imagine that we are analyzing one SNP, and we have these results. The father is heterozygous, and the mother is homozygous, and the fetus is also heterozygous. Obviously, this A must come from the father and belongs to the pathogenic allele, because this is the allele that is shared by both, and this T comes from the mother for the same reason. And also we can analyze the embryos in the same way. Now, one more. Here again, the father is heterozygous, but the fetus is homozygous. So again, one C is from the father, and the other C is from the mother. And for this reason, this G must belong to the healthy allele, because it is not shared with the fetus. And again, we analyze the embryos in a similar way. Now, one more for the mother. Here the mother is heterozygous and the father is homozygous. The fetus is homozygous. So again, this G must come from the father, and this G must come from the mother, and belongs to the pathogenic allele because it is shared with the mother. Again, we analyze the embryos in the same way. And the last example, the father is homozygous, the mother is heterozygous, and here the fetus is heterozygous. So this C belongs to the father, and this A to the mother, and again it is ligated to the pathogenic allele because it is shared between the mother and the fetus. And again we analyze the embryos. So after analyzing of the SNPs in the fetus and in the embryos, we have a situation like this. And now we can compare the alleles in the embryos and the allele in the parents and the fetus. And with this comparison, we can know if the alleles are inherited by the embryos are healthy or pathological. So we can now if the embryos are affected, healthy carriers or fully healthy. Up to now, we were doing this phasing manually for all the SNPs. However, we have developed a web lab platform to do this automatically. Here you can upload all the files from the sequencer, and here you have the results. Here you can see the family trio, and here you can see the results of the embryos. I want to stress out that we are able even to identify an embryo in which we had a recombination. And now, briefly, I want to present a case study. Here we have a couple which is healthy with 2 natural pregnancies and that ended in induced abortion due to a skeletal dysplasia diagnosed by ultrasound. These couples came to our lab for genetic counseling, but in this case genetic counseling was difficult because there is more than 300 genes related to skeletal dysplasia. So we decided to do exome sequencing for the couple and also for to one of the abortions. And we discovered two mutations in this gene, which is related to Jones syndrome. So with this information, we were able to set up a PGD. We were able to set up a PGD cycle. We analyzed up to 7 embryos, and fortunately, we have two euploid embryos that were also healthy carriers. So we have 2 embryos to transfer. One was transferred, and as far as I know, there is an ongoing pregnancy. So finally, we made this solution available to any person interested. We have just launched a kit for PGD that can be combined with the ion reproseq kit. The kit includes all the necessary things for the study of a monogenic disorder, and also access to the online tool for the bioinformatic analysis. So, just to sum up, this is our conclusions about using next generation sequencing for PGD and PGS. We found that the use of NGS is fast and versatile and scalable, since we can analyze few or many embryos. We can combine PGS and PGD in one single test and even the informative test. And finally, it is very cost effective. So, just to finish, I want to thank my team for their hard work developing all the things. Thank you all of you for your attention.
Super. Well, thank you, Fabien and Luis, for informative presentations. We'll now start with the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now just by clicking the green Q&A button at the lower left of the presentation window and type your question into the box and click send. We'll answer as many as we have time for. We're quickly approaching the hour, but let's get started. So there's many questions here, I think, directed to Fabien on Mosaicism, which is obviously a very new and rapidly developing topic. So we have Moreno Lavope from the Genia Laboratory. And the question is, in your opinion, which is the most important factor that could increase mosaicism rate in the lab?
So it's difficult to explain, but there have been some publications showing that inappropriate culture condition could have an impact on mosaicism, which means if the embryo is subjected to some stress during the culture, it could increase the rate of aneuploidy, which means abnormal chromosomal segregation during the first division, and then chromosome in excess on one side of the embryo and chromosome missing on the other side. It should be remembered that early cleavage embryo do not have that many genetical checkpoints, so it's able to go further even if it's carrier of some abnormal chromosomal contents. So if there are some stress on the embryo during the early cleavage, it could be passed through the next cell generation.
Okay, thank you. The next one also is similar. This one is on sort of thresholds here for transfer. Would you transfer a 20% mosaic, and what about 40, 60, 80? So this sort of clinical application here.
Yeah, so we have made our own guidelines based on the published one, and our geneticist will give a different recommendation depending on which chromosome is concerned and whether it's a trisomy or whether it's a monosomy. Obviously, the ones that are high risk of giving rise to an unhealthy child, such as mosaic trisomy 21, should not be transferred, whereas some monosomy which are lethal to the embryo are maybe at less risk, because on the maximum the embryo should rest. But in all cases, in our reproductive centers, when we have no other option, for sure we will transfer first the fully healthy embryo. We'll also suggest to, if there are no normal embryo but only mosaic one, we will suggest to the patient, if possible, to do another IVF cycles. But if the patient would like to transfer for different reasons, we refer them to a genetic counselor to discuss with the patient about the risk, the consequences, and to help them to take their decision for that. But basically our recommendations are based on the type of chromosome involved in mosaicism and whether it's monosomy or trisomy.
If I can add something, in general in my lab, we do not recommend the transfer of mosaic embryos. The only recommendation is, as Fabien said, when this is the last chance for the couple. But if this is done, we always highly recommend to perform prenatal diagnosis with microarrays to detect any possible mosaicism in the fetus.
Sorry, I was on mute. What is the limit in megabases that you've detected for PGS?
Well, in our the experiment we have done, we go down to 17. But it was the way the experiment was designed in the beginning, and it was very accurate. So the question was, would it been feasible to go lower than that? But we haven't done that. I don't know whether at Biography you have any experience on that.
Yes, usually we are able to detect aberrations around 10 megabases, but it is true that it depends on the chromosome affected, because there is some chromosomes that is more easy to detect the aberrations, and others that are more difficult.
Okay, so maybe I'll skip to in the interest of time. Let me skip to this last question here, since it's sort of controversial. And the STAR trial was recently released, which so this is a question from Beatrice at the University of Zurich. Is it clear and demonstrated that PGS can increase pregnancy rates and take-home baby rates? That will be for both our speakers.
So I think we are, as I mentioned, entering in a new area of PGS. It's not that far away that many PGS were done on day 3 embryo or were done with a fish or ray situation analysis. So the question is whether with all those new technology it will be fully convincing, or whether we'll have some disappointment. It's too early to say, but I would say the studies that are coming out very recently are highly positive, and even the most skeptical ones, which have faced the disappointment of the past, might be reassured. But it's, I would say, too early to definitively say that it's the way to go for most of the patients. I don't know.
Well, in my opinion, this also depends on how you measure the success. I mean, if you are taking into account only the duration of having a baby at home, then it is true that maybe PGS increase just slightly the ratio. But if you take into account more things like how many time do you require to have this baby at home, and how many cycles, how many transfers, So you take into account all these things, then PGS is definitively good for IVF.
Excellent. In the interest of time, I think we'll answer the remaining questions via email. Once again, I'd like to thank our speakers, and please note that the presentations will be available on-demand viewing through to April 2018, and you'll receive an email from Labroots letting you know when this webcast will be available for replay. So with that, I'd like to thank our speakers and for everybody who could join us today, and we hope to see you again soon. Thank you very much, and goodbye.
Thank you.